everyone. Uh, I hope I'm audible.
Yes,
Thank
sir.
you for joining for this session of the IAMM Masterclass conducted by the Karnataka chapter of Indian Association of Medical Microbiologists. This is the first session for December. We have been conducting this event since June, and we have successfully conducted two sessions every month. And we thank Dr. Shruthi for accepting our invite, and Dr. Sudhindra has been instrumental in getting her on board. Thanks, Dr. Shruti, for joining. Just to give a brief introduction about Dr. Shruti, she is an alumnus of Raichur Institute of Medical Sciences, where she did her MBBS, and subsequently she's done her MD from JIPMER, MD Microbiology in JIPMER. She's worked as a senior resident in Sanjay Gandhi Postgraduate Institute at Lucknow, and she's also been an assistant professor at Father Muller Medical College, Mangalore. And right now, since November 2022, she's been working as a physician scientist at the Institute of Bioinformatics in ITP, Bangalore. She has numerous publications to her credit, and she also has four intramural and three extramural projects to her CV. She's also won multiple prizes, including the first prize in the quiz in the antimicrobial resistance held in 2019, and she's also won the first prize in NatCon held at Thrissur in 2022. She's been an invited speaker in multiple events, including one at Nepal. And she's also talked on biomedical waste. She's also talked on medical mycology. So she has varied interest in mycology, in microbiology. And apart from that, she's also been an editorial board member and a reviewer for many journals. So, I mean, this is in brief about Dr. Shruti, and very hearty welcome to you, Dr. Shruti, and the stage is yours for discussion. Over to you, Dr. Shruti.
So I can start presenting, sir?
Yeah, sure, please.
Yeah, thank you. Okay, please let me know if the slides are not in slide share mode. I've shared now. Good evening, everybody. Today I'll be talking about molecular beacons in diagnostic microbiology. Basically, I'll be talking about the concept of what is a molecular beacon and how do we use that and how can we use that in the future for the diagnostics, because we know there are different kind of probes. Sorry for the disturbance. Okay. So yeah, I'll be covering the topic under these headings: introduction to molecular beacons, structure of molecular beacon, and how do we design the molecular beacon, what is the mechanism of action, what is the principle of threat, and the types of molecular beacon and design consideration, applications in microbiology, gene export, strength and advantages, limitations and emerging applications. So what is a molecular beacons? These molecular beacons are nothing but single-stranded oligonucleotide probes which are designed in a hairpin structure, as you can see from this figure. You might have come across different kind of probes which are currently used in PCR, like Tachman probes, molecular beacon probes, LNC probes, degenerate probes. So these are different kind of probes. One of such probe which is of important use in microbiology and which we need to know in microbiology is molecular beacon probes. So they have a stem and a loop structure. This loop structure usually is complementary to the target sequence. I will tell you where do we not take the exact complement when we are designing the molecular beacon for. Sorry, I just have to mute this one call. I'm really sorry for this. Yes, yeah. So these molecular beacons, they are labeled with fluorophore on 5' end and quencher on the 3' end. So as similar to any other kind of probes. The fluorophore and quencher, they are held together close to each other, which doesn't have any or very low background because of a mechanism of FRET. I'll explain you in the future slides, in the subsequent slides, what is this FRET and how does the fluorophore, when it is present and when the conditions are there, still it will not fluoresce when the target sequence is not available. So, and just like any other PCR, even these fluorescence will be emitted. I don't know. I'm really sorry for this
Sir,
one
the
thing.
speaker's
I'm
audio
getting
is breaking.
back-to-back calls. I'll just um,
Sir, the speaker's voice is breaking.
Yeah, I think she's received a call, I guess. I think she'll be back in a moment, sir.
hello, sir. Am I audible now?
Yes, Dr. Shruti, please
I'm
go ahead.
really sorry for that call. I don't know who was
Mm,
that. They were calling me back to back, so I had to pick that call.
No, no,
I'm
no
really
issue, no issue. Please go ahead.
okay. So I'll go from the second slide onwards. Yeah, I had already explained, just like any other probes, these molecular beacon probes also have a fluorophore and a quencher. And when there is an emission, you see the fluorescence, which is similar to any other probes which we find in any of the type of PCR. So how are these probes different compared to Tachman probes? In this class, I will be briefly touching upon the applications of these probes, but I'll mainly be focusing works. So when you go out of the class, you should be confident enough to design a probe on your own, and you should be able to do a PCR on your own. Okay, so coming to the structure of molecular beacons, they have loops which are complementary to the target sequence. They usually contain 18 to 30 base pairs in case of classic molecular beacons, and around 20 to 24 to 30 base pairs in case of sloppy molecular beacons. So again, I'll tell you why is it called sloppy molecular beacon, and the classic one is the regular molecular beacon, which was first designed. And then, as I already explained, the stem are the self-complementary arms, which you can see from this figure. If my screen arrow is visible, or I'll just use the pointer. Yes, as you can see from this figure, this is a stem structure which has taken from here. This stem structure has a self-complementary sequence. That means G is complementary with C and A is complementary with T, G with triple bond and A with double bond. So we usually have a stem structure which is typically GC rich. Why do we have a GC rich stem? So that it ensures the stability, and stem doesn't open all the time, and it needs a particular melting temperature to open. Okay, so then moving forward, I already explained these stay as a hairpin loop structure or the closed hairpin structure in the absence of perfect complementary target. If there is a particular mutation, they will not bind to that particular mutation. Even if it binds, it slips off and it stays as a hairpin loop structure. So it is more sensitive, more specific, not sensitive, more specific as compared to Tachman probe if we are using a classic molecular beacon. Please keep this in mind. It's a classic molecular beacon. Okay. So then, as I explained, every stem and a loop structure will have a particular TM, that is a melting temperature. So we know there are three basic steps in PCR. One is your denaturation, where the double strand will become a single strand, and then you have annealing, where it goes and binds to the target sequence, and there is an extension. In the extension phase, your polymerase binds and it will complete the replication process. Yeah. So when do we need this melting temperature? When you have a annealing step where we put certain temperature for annealing. If you have designed, or if you have just programmed a PCR machine, you know what is the annealing temperature for this PCR and how much annealing temperature has to be kept for this. Even if you are doing conventional PCR, you set the annealing temperature or the this thing based on the primer TM, whatever is given. And in case of molecular beacons, again you set the annealing temperature based on the TM provided for the loop structure and the stem structure. The stem structure, what we design, these are not what to say, this is not a hard and fast rule. You need to have GAGCC as a stem structure, any sequence of your choice, which has to be 5 to 7 base pairs, and they have to be GC rich. Okay. And how do we check whether particular stem structure you have chosen is suitable for your loop structure or not? For that, if you go back to your basics of molecular biology, you know that your G has a 3 bond, G and C, and for the A, you have double bond. So if there is a triple bond, there is a certain amount of energy which is needed to break that bond. And if there is a double bond, there is a certain amount of energy which is needed to break that bond. So this stem TM should be lower than the probe target duplex. When this probe binds, as you can see from this figure, when the probe loop structure binds on the target sequence, there is a melting temperature. That melting temperature and the stem melting temperature should be at least -5 to 10 degrees apart. That means your stem melting temperature should be less than the melting temperature of your target duplex, target probe duplex. Okay. Fine. So I'll move to the next slide. So how do we design the molecular beacon? Just now I explained what is the structure of a molecular beacon. So we'll take an example sequence. This is a dummy sequence. This doesn't match anywhere. Maybe it might match with some complementarity. Otherwise, it's a dummy sequence which I have made up. So this is the target sequence for which if you want to design a probe. Okay. So you have 5' fluorescent end and a 3' BHQ end. That is black hole quencher. That is a quencher end. So again, when you are tagging the fluorescence, why do we tag it in the 5' end? Why not in the 3' end? Again, these are all the basics of molecular biology, which we keep reading when we read any kind of probes. So here in the molecular beacon probe, I have given a stem structure of CGC GTA, which is highlighted in red. And this is the structure which is complementary to your target sequence. And then you have another complementary pair for the stem structure and your BHQ, that is black hole quencher. Okay, so in the basics of molecular biology, we talk about something called Valle's rule. That is the TM calculation for any of the DNA strands. So the loop has here you can count, the loop has a 17 nucleotide. When you count the bases, A was around 6 bases were A, around 5 bases were T, around 3 bases were G, and around 3 bases were C. And it was giving an approximate TM of 46 degrees centigrade. How did we calculate that TM? Based on this Valle's rule formula, where 2 degrees centigrade into, it is n number of A plus n number of T plus 4 degrees centigrade into n number of G plus n number of C. This is the Valle's rule, which we basically calculate. But practically, none of us usually sit and do this kind of things. Okay. In the lab, because when we are doing multiplexing, it's practically not possible to sit and count each basis and how much it will come. But when we are designing a probe, 1 or 2 probes, then definitely we can do this by calculating the stem TM. What is the loop TM? We calculate in a way that it will fit to our probe which we are designing. Okay, so the stem TM, as I already explained, should be much lower than the loop TM. And the beacon hairpin will open readily only then, and it hybridizes to the target. This is a common or a basic principle which we have to keep in mind. Okay. And so as I already explained, when we calculate the TM, we calculate using some of the reagents which we use, like the buffer pH and the salt concentration, magnesium concentration, and the oligo concentration. Oligo concentration is nothing but when we are adding primers, we always mention, no, I added 0.3 micromolar primer, or I added, and I took around 0.3 microliter from the 100 millimolar primer. So we mention usually how much concentration of primer we are adding. So in the same way, you have to mention how much concentration of probe you are going to take for your particular PCR, and using that, using a nearest neighbor TM calculators. The common one which we use is IDT Oligo Analyzer, which is a free software again, where you can put your buffer conditions, put your salt concentrations, magnesium concentration, oligo concentration which you're going to take, and see the accurate TM values for your particular reaction, which you can go back and try after this class. So just now I explained about the designing of a probe. When you the basic designing, how we start is we download the FASTA sequence where we want to design the probe for, and we mark it in, and then we upload it in particular designer softwares are there. Like how for primer designing you have Primer Plus, Primer 3 Plus. We have a particular software which we use for designing molecular beacons, and it is called as Beacon Designer. So in the Beacon Designer, once we upload the FASTA sequence, it will give us nearby matches what we can use for the probe sequence. Again, with the nearby matches, you can say, okay, I want to sequence from this end to this end, and I want to have a probe of maybe we always not take like it is the range is 15 to 30. We always don't take such a long probe. We sometimes keep a small probe also when you want to do the reaction fast. Small probe and small TM, small stem. Okay. So we can identify where we want to match it, and we can design our own stem structure and put it in the IDT oligo analyzer. It will tell you what is the TM, accurate TM with your buffer conditions. Okay, so once you design the probe, you should know how it is actually acting, right? Then only you will be able to able to program your PCR machine at what stage the capture should be there. Because as you know, when we are programming a RT-PCR machine, we click something called camera image, which is seen, no? So that is the capture image. Where you want to capture the fluorescence is also important in case of the type of probes you are using. Okay, so the mechanism of action, I'll give brief steps which will be useful for you to also write in the exams. The first step which happens is stem melting and beacon opening. Once the target binds, once okay, well, let's walk you through the steps. So once you start your PCR, the first stage which happens is denaturation. So denaturation means it has to separate from each other, right? So once the hairpin opens and it goes and binds to the target sequence, there is a conformational change. That means the closed hairpin gets opened and becomes a linear form. So that is the first step. Stem has to melt and the beacon has to open to become a linear form. Unlike Tachman probes, which is already a linear probe, right? So then we have target recognition by this loop sequence. As I already mentioned, the loop sequence is the complementary sequence for the target. Okay. So when there is a correct target sequence which is present, it has to hybridize to the loop due to higher thermodynamic stability. This will stay back. This we have to keep in mind. That's the reason why we always mention the loop TM should be more than the TM of the stem. That is when it will stay back. If there is a single mismatch in case of classic molecular beacons, this thermodynamic stability won't be maintained and the probe will fall off. Okay, so you might get a false negative in those cases if the patient is having particular mutation. In case of classic molecular beacon, please note that word classic. Okay, so then the third step is separation of fluorophore and quencher. So once the hairpin is opened, it increases the distance between the fluorophore and quencher. As you have seen in the previous picture, both the stems were together and the fluorophore and quencher were together. So there was no fluorescence which was seen, or no fluorescence which was absorbed or identified by your machine. Okay, so this separation of fluorophore and quencher after the opening of the hairpin structure to a linear form actually causes the fluorescence, and fluorescent intensity is directly proportional to the amount of target hybridization, which is true to any PCR, be it conventional or be it RT-PCR, where you are adding particular amount of DNA or particular nanogram of DNA. You have 10 power 6 copies. You'll more fluorescence. You have 10 power 4 copies, you'll have less fluorescence. Coming of fluorescence. How is this happening? This is happening with the principle of FRET. You might have studied FRET as fluorescence resonance energy transfer in most of our textbooks, because even I studied that way during my MD. But later I realized it's based on the principle of faster resonance energy transfer, because this quenching is not based on fluorescence. This quenching is based on dipole-dipole coupling. Okay, so it is called as faster resonance energy transfer because it was first invented by this scientist called as so as the fluorescence is separated from the quencher, the fluorescence is emitted. That is the basics of FRET principle. So I'll explain you very briefly how the Tachman probes and molecular beacon probes are different. So the Tachman probes are hydrolysis probes. Once as you can see from this picture, Tachman probes are the hydrolysis probes. This white thing is your forward primer, and this maroon thing is your reverse primer, and you have your probe here binding. Okay, so once your probe here binding, which is orange in color. So once your primer binds, your Tach polymerase, which you use, will start extending it during the extension phase. Your probe is also binding in certain position. So once it is extending, it will chew off the fluorescent dye which is attached on the 5' end of the probe. Once it gets chewed off, so there is free fluorescent dye lying in your PCR machine, which will keep giving the fluorescence because the quenching is lost by the chewing of fluorescent dye from the probe, right? So what happens in molecular beacons probe? That's the reason why we call it as hydrolysis probes. So probe is cleaved during the extension, which causes a release. That's why if you go back and observe department, you can extension phase, right? So and also once it gets chewed off, there will not be any quenching. So the signal is permanent and it gets accumulated each cycle, right? So then we have molecular beacon probes. I told you these are hybridization probes. So basically what happens, when once there is a denaturation, your probe opens to become a linear form, right? And it goes and binds when there is an exact complementarity. So after it binds, here both the probes and the so technically there should be fluorescence which can be measured, right? So what is this step where it is going and binding? This step is called as annealing step. So here the capture is always done during annealing step, right? So as I already mentioned, denaturation melts the stem temporarily. It's not a permanent melt, because what is happening, our polymerase here, what we use is not a Tach polymerase, because Tach polymerase has something called as 5' to 3' exonuclease activity. Again, don't get confused between 5' to 3' exonuclease activity and 3' to 5' exonuclease activity. The 3' to 5' activity is basically for proofreading. When once your Tach polymerase completes the one full stretch of extension, it goes back and see was there any mismatch. So that is your 3' to 5' one, which is for proofreading. Don't get confused with 3' to 5' activity. Here I'm particularly mentioning it is 5' to 3' exonuclease activity should not be there. That means when it is moving from left to right, as you can see from here, from 5' to 3', it should not be cleaving your fluorophore. So when you are using molecular beacon probes, we use a certain kind of polymerase, which is we call it as exo- polymerase. One of the important or the common examples which we use, and the cheaper one is Vent polymerase. Okay. So why do we use this kind of polymerase? This is one of the difference which you have to keep in mind. Other than when the signal is captured or what kind of probe, you should also remember what kind of polymerase is used, right? So why do we use this polymerase? Because we don't want it to cleave the probe. We don't want the fluorescent dye to be cleaved away from the probe. Once it gets cleaved away, the continuous emission will be there. So then you might get a lot of backgrounds in that. So never use a Taq polymerase for molecular beacons. So the signal stability is reversible on and off during each cycle I have mentioned. That means what was happening in Tachman probe, your fluorescent dye was getting cleaved away. So it was cleaved away from the probe. So it was always there during the cycle, and every cycle it was getting accumulated from all the probes. But what happens in molecular beacon? First cycle, then again it will bind, because one beacon will go back to its thermodynamically stable structure. What is the thermodynamically stable structure? That is hairpin structure. So you will not have fluorescence in other phases usually. So that's the reason why we say signal is always reversible on and off each cycle. Okay, fine. I hope you understood what are the differences between the regular Tachman, what are the different things you have to do when you're using Tachman probes, what are the things you have to use when you're doing molecular beacon probes, when you have to capture the fluorescence, what kind of polymerase to be used. Okay, so I was talking about the main principle, that is FRET, faster resonance energy transfer. This is nothing but the transfer of energy from one fluorescent molecule to another, that is from the donor to the acceptor, without emitting the light. This is a catch here. Here the excitation is happening, as you can see through in the pointer which I'm pointing. So this purple and the blue wavelengths are for the donor molecule. So excitation wavelength will be different, and it will be usually less than the emission wavelength. This is the basics which we have learned during our physics classes. So what happens? This FRET transfer is without emitting light, and it is through dipole-dipole coupling. So once there is an excitation by the light source, there is an emission also has to happen, right? So if both the donor and the quencher are kept within a close distance of less than 10 nanometer, there will not be any emission, there will be quenching even upon excitation. Why? That is because of the spectrum overlap, which you can see. Like, you cannot randomly use some donor for some acceptor. There will always be a donor acceptor pair based on the wavelengths. And also the wavelengths you have to choose for your PCR machines will be different. They have a different filter and all. So that is again a second different part of it. But here, when you're choosing the donor and the acceptor, you have to choose particular donor and acceptor whose spectrums are overlapping. The emission spectrum of the donor has to overlap with the excitation spectrum of the donor. Okay, so these are the key requirements when you are using the choosing the donor and the acceptor fluorophores. Okay, and the donor has to be excited by a light source. These are the three key requirements which we usually mention. So what is this dipole-dipole coupling? It is nothing but when the fluorescent molecule gets excited, there is an oscillating electromagnetic field which is generated around this. And when the two molecules are less than 10 nanometers apart, this oscillating, this is too much physics, but yeah, this oscillating electromagnetic field will be preventing the fluorescent from getting excited. Okay, fine. So this separation of dye causes the loss of FRET, usually when the beacon opens or when the cleavage happens in the Tachman probe, and the donor fluorescence will increase. Okay, so this is the basic principle used in treatment by the PCR machines. Okay, till now, which has to have a perfect. I'll give you an example.
There is some voice breakage in between. Can you please check the network connections?
Yes, sir. Is it okay, sir, now?
Hopefully, it should continue.
Okay, sir. If there is any problem, I can record and send also later.
Recording is on.
Yeah, okay, sir. Okay, sure, sir. Till now I was explaining about classic molecular beacons. Now I'll explain you about sloppy molecular beacons. Those classic molecular beacons were of perfect complementarity to the target sequence, right? I was telling, if there is single mismatch, the probe will fall off. Unlike your Tachman probe, which might accommodate small mismatches, it will not be very specific like that. But in case of sloppy molecular beacon, why did they design the sloppy molecular beacon? Because they wanted something which will not give lot of background fluorescence, meanwhile which will not also fall off if there is some one or two variants or mutations. So that's the background how they came up with sloppy molecular beacon design. uh, and what they have done in sloppy molecular beacon means, as the name suggests, it is sloppy, it is not very strict. That means they have designed a weak stem, as you can see from here. The deliberate mismatches in the loop, as you can see from here, one mismatch was added. Mismatch means the target complementary to the target sequence was supposed to be GGATGC, but this deliberate mismatch was added so that we wanted some of the loop to be hanging loose, so that we get a particular TM differences or TM shifts when we are designing the probe to detect the mutations. For example, this is the variant which I want to detect. I have designed my sloppiness or the engineered mismatch very far away from this. Here it might not look far away, but when we are designing, we usually design quite far away. We design around 25 to 26 base pair probes. Okay, so we design them quite far away. We want them to go and attach. Meanwhile, we don't want them to fall off, and we want them to identify the mismatch. For example, here you can see my variant at question is A. So it was supposed to be A in wild type, but in case of, for example, if you take humans, it might come as G or C or T, because humans have two pairs of chromosome, two different alleles can be there. Both the alleles can be same also, both the alleles can be different also. So, or both the alleles can be a variant also. Wild type means which we usually take a standard GRCH38 for human, and for MTB it is H37RV, the genome which we download from the NCBI database. So that is the wild type. Opposite to that, it can be T, G, or C, which can anything can be there in your detection range. So we want to detect be it whatever mutation is there. So we have this is our targeted question, and this is our deliberately engineered mismatch, and this is the deliberately engineered sloppy stem. We want the stem to be weak and single loop mismatch to make a controlled sloppiness. That's the reason why we call it as sloppy molecular beacon. Okay, fine. So as I already mentioned, we want both the wild type and mutant to still hybridize, but with different TM values. It's not that I just want to know whether it is positive or negative. No, I want to know if it is positive, if the patient is having a variant or a mismatch, he is having a resistance. I really want to know he is having a resistance. I want to have a signal for resistance. What was happening with classic molecular beacon probes was, if there is a wild type, it was binding, we were getting fluorescence, it is positive. If it is not binding, we were taking it as negative. No signal was meaning negative. But how do you say if the PCR happened, you will have controls, positive control, negative control and all, but still, how do we confirmly say, okay, this is the particular mutation which is there, which is very important when we talk about genotyping in case of patients. We want to know what allele is there in which strand, whether he is having both the wild type alleles or both alternate alleles, one alternate or one wild type. That can be done only when you have a sloppy molecular beacon kind of probes, where you get different TM values for wild type and different TM values for mutant and different TM values when it is heterozygote. Heterozygote means both mutant population and wild type population is there. How do we match this for microbiology? When you have heteroresistance population, you have, for example, 50% of the strains were sensitive for rifampicin and 50% of them were resistant for rifampicin. You will design two different probes, and multiple probes you have to keep designing for the same sample if you go with classic molecular beacon. as I mentioned, you can very well design a probe for even mutant variant also, right? Which is practically not possible. You cannot keep designing multiple probes for multiple mutations. You want all the work to be done by single probe. So that is where this sloppy molecular beacon design works. And the ideal TM difference, which we usually mention for measuring genotyping, is 2 to 6 degree centigrade. Okay, so this I've already mentioned while talking. I'll just rush through this. So the requirement in molecular beacon, sloppy molecular beacon, is hairpin should be loose, but we don't want the background to be high. So fret is needed there when you don't want the background to be very high. And we want distant melting peaks for the variant and the wild type both, only when the fret mechanism is there, and when you do high resolution melt curve analysis, only then you can get the distinct peaks for wild type and the mutant. Okay, and we wanted a highly sensitive probe which will not get degraded, or which will, the stem doesn't just open off and doesn't bind properly. So in these places, again, we wanted sloppy molecular beacon. And the most important thing what is currently needed in microbiology is multiplexing multiple things in a single reaction. We want results fast because patient don't wait, clinicians don't wait, right? So for the multiplexing, rather than classic molecular beacons and having different kind of maximum how many dyes can you add in one PCR machine? Not everybody will have QS5. Few people might have a normal rotor gene RT-PCR machine as well, right? So a qPCR machine as well, right? So your multiplexing ability will come down when you have less channels in your PCR machine, which can be addressed using your sloppy molecular beacon, where 3 different things can be identified the single probe, and you can have multiple probes like that, around 5 probes and 5 different fluorophores. Still you can identify 15 different variation in a single reaction, right? That's good, right? Yeah. So then I was talking about high resolution melt curve analysis. So it is nothing but a post PCR analysis method that detects the differences in the DNA sequence by analyzing how the double-stranded DNA is denaturing as the temperature increases. Okay, so the basic principle of HRM is nothing but every DNA has a unique melting behavior. GC has a higher melting behavior because of triple bond. AT has a lower melting behavior because of double bond. And even a single nucleotide change or a mutation can shift this melting temperature. As you can see from this figure, this is basically one of the genotyping assay. So here you can see the patient was supposed to have AC and AC. If patient is having AC and AC, haplotypes means 2 different things. The patient humans are having usually diplotypes. That means they have a diploid chromosome and they will have 2 different alleles on each strand. So this basically talks about one strand. This basically talks about another strand. Again, yeah. So if the patient is having both AC on both the strand, that is, he is having a wild type. Okay, so if the patient is having AC, you get this green color peak. Fine. So if the patient is having both alternate, you get a different peak. Yes, because AT melting is different and the CG melting is different. Fine. So if the patient is having both AC and AT, what do we get? So he is having a AC and he is having a AT. Both the peaks will come if the patient is heterozygote. How it holds good for us microbiologists if we are not sequencing humans, if we are not doing pharmacogenomics or something. So it holds good for us when we talk about the different mutations or the mutations present in your, for example, the most common one, which I will be explaining in detail, rifampicin, your gene expert. Okay. So these are the key features. I'll just rush through the key features of this, because there are different types of molecular beacons and not everything is used widely. The most common ones which are used widely are classic molecular beacon, sloppy molecular beacon, and multiplex molecular beacons, right? So the dual stem molecular beacons, Wobble molecular beacons, these are usually used with CRISPR related studies, and mostly they are used in research purposes, not in the diagnostic assays. So but you can know what are these different kind of molecular beacons. There are 7 different types of molecular beacons, and each have a perfect key feature and the use case. Okay, so the classic molecular beacon, they have a perfect complementarity, which I kept on saying, and they can be used for detection, SNP detection, or quantitation in qPCR, right? And then SNB, the key feature is we have a intentional engineered mismatches, and this can be used for mutation scanning and high resolution melt curve analysis. Okay, then we have multiplex molecular beacons where you can use different fluorophores and multi-target assays can be done. And then you have dual stem molecular beacons when you want extra stability, when you have longer targets which you want to capture, then we use these dual stem molecular beacons. You want to capture longer targets, but you cannot keep your fluorescence and the acceptor very far off. Then we give 2 different stems for that molecular beacon. And then we have wobble molecular beacons where we use something called as degenerate bases. This degenerate bases, uh, this currently is a trending thing, where this degenerative basis you can use when you have particular sequence which is not too different from each other, especially when we talk about strain typing. You want to know which strain came first in case of transmission related studies and all. There these wobble molecular beacons are used. And then we have truncated molecular beacon, which I had already mentioned previously. You might want to design something which opens faster and binds faster. You want to have such kind of thing where you are not increasing the temperature much and all. Then you can have a short loop and short stem kind of molecular beacons for faster hybridization. And then you have a universal design. Again, you have something called universal primers, universal probes and all. When you have a universal design for high throughput for barcoding and all, then we can use this zip code beacons. So currently, the 2 important things for microbiologists or which have application in microbiology diagnostics are molecular beacons and sloppy molecular beacons, apart from your regular Tachman probe. This table might be useful for you to quickly revise during the exams when you don't have time and still you want to just revise some points. So this, all of you know, it's a linear structure for Tachman, and it's a stem and loop structure for molecular beacon, and it has intentional mismatches in case of sloppy molecular beacon. And the signal mechanism is by hydrolysis. For a Tachman probe, for a molecular beacon and sloppy molecular beacon, it is hybridization. And the fluorescence is captured during extension phase in case of Tachman and during annealing phase for both MBs. And the specificity is high for Tachman. Again, please remember this point. People usually get confused with the 5 prime 3 prime part, and because we ourselves used to get confused when we didn't know the exact mechanism how it works. And your Tachman can actually tolerate some mismatches, and but your molecular beacon, the classic molecular beacon, is having higher specificity. It will not tolerate mismatches. So I will, if possible, people can guess in the chat where this classic molecular beacon is used in microbiology and where the sloppy molecular beacon is used. In subsequent slides, I'll tell. Okay. And the probe integrity after cycle, it is destroyed in case of Tachman, whereas it will be preserved in case of both the molecular beacons. And the Tach polymerase you can use in case of Tachman, but you have to use a polymerase which doesn't have 5 prime to 3 prime exonuclease activity in case of both molecular beacons. Okay, so the best use case for quantification you can use in Tachman, and for SNP genotyping or single based discrimination, when you want to do, you can use molecular beacons, and when you want to detect a lot of beacons. And the TM designing, again, basic principles of designing, you have to remember how much difference should be there between the same TM and the target TM for the molecular beacons. Fine. Yeah, let's go for what are the applications of molecular beacon. So we learned about what is the molecular beacon, how do we design that, what are the types of molecular beacon, how is it different compared to Tachman probes, so all those things. So what are the applications which are relevant to microbiology? I'll be talking about that. So for real-time PCR diagnostics, especially for TB drug resistant assays, And when you want to do viral load monitoring, you can use these molecular beacons. And for pathogen identification and genotyping, this might sound almost similar to Tachman probe ka bhi, same uses people are telling. For molecular also the same uses they are telling. Again, it differs from organism to organism what you want to see in that organism, whether it has particularly only one mutation or it has the entire mutation catalog, how you get in your MTB. MTB, WHO mutation catalog, if you see, there are so many mutations, around 50 to 100. To detect SNPs, as I mentioned, you can detect the SNPs single variant mismatch, or you can also do genotyping where you can say, okay, this particular patient is having two wild type or two alternate, or one wild type, one alternate. And when you want to do allele discrimination, the same thing. And for gene expression analysis. Okay, so molecular beacon in GeneXpert, this is the most common use case in microbiology which you need to know. So in GeneXpert module, you might know there are two types of GeneXpert module. One is G4, which was your earlier one, GeneXpert MTB-RIF, and then you have Ultra. Okay, so in the G4 MTB-RIF, they had 5 molecular beacons from A to E. They had labeled it as A to E. And these designs were perfectly matched with the wild type RPOB sequences. So what was happening? It could able to detect there was a presence of M. tuberculosis and presence or absence of rifampicin resistant mutation. It was not able to say ki, okay, like multiple mutations could not be picked by it. So what was happening? If there is a wild type sequence, all the 5 beacons will fluoresce normally. If there is any mutation in any range, corresponding beacon will fail and it will be reported as RIF resistant. So this classic beacon, as you know, it usually works on and off mode. If probe fails, there is no fluorescence, so it will give a binary interpretation as plus or minus, which is not the case, because sometimes you might be having some mutations which are of no significance. If you read that WHO mutation catalog, where they have classified the mutations into associated with resistance interim, not associated with resistance. So there are different classification again you have for the mutation catalog. Even though mutation is there, that might not be really affecting or causing the resistance in the population. So this is nothing but a schematic representation of that RRDR region, RPOB, which is a 81 base pair resistant determining region, where they had designed 5 different probes. So this was happening. So the sensitivity and specificity of GeneXpert G4 version was much lesser than the current GeneXpert version, which is GeneXpert MTB-RIF Ultra. So in MTB-RIF Ultra, they've included much more mutations and with only 4 probes. Here, how they are working, how they are identifying the mutations is based on the melt curve analysis, which I already mentioned. So what is the difference here? In the GeneXpert Ultra, they have used sloppy molecular beacon probes for detecting RPOB RRDR region. So here the Ultra replaces the classic exact match beacons with the sloppy molecular beacons. This image you can see, don't go by these numbers, 428 and all. In the behind table it was 521 and all. Again here one more mismatch which we had ignored even during our MD time, was these numbers are still given based on E. coli numbering code, and the number 521 and all is given based on the MTB numbering code, something like that. So here what you can remember is, sorry, it's vice versa. So what you can remember is, here, though they have designed this sloppy molecular beacon probes, they have designed it to be overlapping to each other. What you can see here in blue, though it is overlapping probe, these are usually not picked up by RPOB1 probe, but these these particular mutations are picked up by the RPOB2 probe or the second probe. Whereas this particular mutation, doesn't get picked up by RPUV1 probe, but it gets picked up by it doesn't get picked up by RPUV2 probe, but it gets picked up by RPUV1 probe. So that is what though the probes are overlapping on each other, what gets picked up by what is based on the how the TM difference they have designed. It's not that it is not binding, it is binding, but the TM difference cannot tell it apart. That's the only reason, right? Okay. So these SMBs are designed to hybridize over a broader sequence window, and they produce a distinct TM signature. As I already mentioned, for wild type there is a different TM, and for mutant there is a different TM, rather than that classic readout like by either it is binding or no binding. Okay. So, and Ultra uses 4 probes, which I already explained, and these produce a measurable TM shifts. So this image I have taken from Dr. Samitesh Chakravarti's paper. He's the one who has designed Gene Expert Ultra probes. So basically, as you can see from this picture, if there is a wild type, so they will have these particular 4 different melt peaks. If the person is having mutation in the region where RPOB is binding, there is a melt curve shift. It is supposed to come at 71, but it is shifted to 63. So the corresponding melt shifts means corresponding mutation. Again, those are given in the Gene Expert Ultra paper, which I've put in the reference, which you can go back and read how much melt shift will be there in which probe for which mutation, and all those are given. Okay. So, as I already mentioned, SMB Ultra probe, they hybridize and then melt. And this TM peak position records the shift pattern to classify the wild type versus the specific mutant signatures. So, this is one of the very important use case of classic molecular beacon and sloppy molecular beacon, which every microbiologist should know and remember. Okay. So, other than that, there were various other uses. So, what are the strengths and limitations of this? The strength, it has high specificity if it is a classic molecular beacon, excellent single nucleotide discrimination, and very low background. The stem keeps fluorophore and the quencher closer. So that's the reason why it will have very low background as compared to other probes. And it gives a reversible on and off signaling, unlike the continuous signaling which you get in Tachman probes, which is ideal for a melt curve analysis. Okay. So, and then it has real-time detection without probe degradation, and it is good for short targets like miRNA mutations, usually most commonly used in cancer diagnostics, and drug-resistant codons. And it is compatible with isothermal assays. These are again newer emerging technologies where molecular beacons are used along with the existing technologies or the newer technologies, LAMP and CRISPR readouts. And it is multiplex friendly with different fluorophores. What are the limitations? It has a very complex design requirements. And when you are designing, so usually when we are designing the primers and probes, so we design it with intuition most of the time, though there is primer plus and all those things, we design it most of the time with intuition. So this might work, and then we blast and see. So it involves a complex design requirement where not always your intuition will work, where you have to again go back and check in your softwares whether it is good pair or not, with putting all your conditions into picture. And then it has slower hybridization kinetics because it has to open from a hairpin loop to a linear probe, unlike Tachman, which is already a linear probe. It is sensitive to target secondary structure. So what happens if you're designing the stem structure, if you have a lot of G and C contents, you should make sure it is not forming a secondary structure or a quaternary structure where all the 4 Gs are clumped together. So again, you have to keep those things in your mind. Don't put all the 4 or 3 Gs together. Have a in between, keep a or a T or a C, A or a T. So you don't get those G quadruplexes when you are designing. And it is not compatible with TAC exonuclease activity. You need to buy Vent or Exo, which are again a little bit more costlier than your regular TAC polymerase. Then the signal can be measurable only during annealing and not extension phase. So the potential background at high temperatures, if the stem is very weak, this also you have to keep in mind when you are creating intentional mismatches and a weaker stem you are forming. Make sure that it is not too weak to give you background fluorescence. And Multiplexing requires careful spectral planning. If you are going to put 5 different fluorophores, make sure you are using particular specific quenchers for that, and it is not overlapping with each other. So there are some emerging applications, which is one of my interest area as well. It can be used with digital PCR, it can be used with CRISPR-Cas diagnostics. All of these are currently in research stage. None of them are in your current diagnostic applications. So you can just remember it can be used along with them. So this super resolution microscopy is nothing but they are putting their molecular beacons for differentiating things, which current use case I have not yet found for our microbiology stuff. Then for nanopore sequencing enrichment, now nanopore doesn't stay like a closed platform, unlike earlier where you had to use everything from nanopore. Except for the barcode, you can use many things from different companies, and you can still do away with your sequencing. Sequencing is not very complex, it's nothing but your PCR. And then molecular computing, we recently had a meeting with one of the companies called Algorithmics Biologics. They do molecular computing, identify how you can do massive multiplexing, like 250 reactions in one single thing, one single PCR and all. So yeah, that is a emerging field. Anybody who's interested in mathematics and molecular biology together can look for this. Then mRNA biomarker detection, nanomaterial enhanced biosensing, and lamp-based assays where molecular beacons are used. These are some of the other emerging applications. These are the references. These papers you can go back and read how nicely they have explained how they have designed the probes for GeneXpert and MTB-RIF Ultra and all. So thank you for your attention. I'm open for questions. I'm really sorry for the unavoidable disturbances happened during the presentation. Thank you.
Thank you, Dr. Shruti, for a wonderful session. I think it was very enlightening for all of us, and I can see that we have very eminent microbiologists like Dr. Appal Raju sir, Dr. Jayanti Savio, Dr. R.D. Kulkarni, who had joined us during the session. And yeah, we were all in rapt attention throughout. So thank you for a very wonderful session. And if there are any queries from the participants, I mean, I'm sure Dr. Shruti would be happy to answer them. Just a snippet. I mean, Dr. Shruti is one of those whom Dr. Sudhindra has been highlighting as a hidden gem in microbiology, and I'm sure all of us can make out why.
And so can we request Dr. Appalaju sir to give some comments? Dr. Appalaju sir, R.D. Kulkarni.
R.D. Kulkarni sir and Appal Raju sir, any comments, sir? In the chat, I can only see compliments. Wonderful as always, and thank you for enlightening the presentation. If there are any other queries, I'm sure Dr. Shruti will be happy to answer them.
Hey,
And on behalf of Indian Association of
hey,
Medical Microbiologists Karnataka chapter,
sir, just a minute, sir.
yeah, please.
Hey, can I request Dr. Shruti to give a small brief introduction about the Institute of Bioinformatics and because many PG students have been asking about what is this Institute of Bioinformatics and what is the exact work.
Yeah, sure, sir. Basically, Institute of Bioinformatics is a misnomer, I would say. We don't just do bioinformatics. We are actually known throughout India for proteomics approach because my co-guide, Dr. Akhilesh Pandey, he is a faculty at Mayo Clinic. He is the founder of Institute of Bioinformatics. So, from India, if we talk about Human Genome Project and all, so the human proteome database or the human proteome map was done from India, from Institute of Bioinformatics. And in Institute of Bioinformatics, we have a physician scientist post where the person who has done MD and who is very much interested to do research, be it a proteomics field, metabolomics, lipidomics, or genomics field. We currently have around 5 physician scientists. I'm the only microbiologist. Rest everybody is from biochemistry background. So there is an option for applying, and you have to clear the interview, and you can join for PhD. Basically, I'm working as physician scientist, also doing my PhD. So here in Institute of Bioinformatics, I work on pharmacogenomics of anti-TB drugs, and I also work on how to design different sequencing panels, basically nanopore-based sequencing panels, and how to design different kind of how to do high number of multiplexing. I have currently multiplexed up to 42. I can sequence all the 42 genes in a single PCR run. I can put all 42 genes in a single PCR tube. So that is what we have done. My guide is Dr. Renu Verma. She is alumni from Stanford University. Other than that, we work on cancer genomics. We work on genetic diseases. So in infectious disease part, yeah, we do a lot of MTB genomics and diagnostic assay development, and pharmacogenomics is my main topic for research, where I do therapeutic drug monitoring and sequencing human genome, and developing a panel for how to identify the ADR. When the patient comes to the clinic, you should be able to say, okay, he's having TB, he's having this drug resistance, okay, you have to give this particular drug in this particular dose. So that is what is our broader umbrella on which we are working. So our lab is called as Infectious Disease and Precision Medicine Lab. So our results will come out in a few years. Currently, we are still enrolling patients and doing the lab work. Panels are developed, validated. We have to do external validation. So those things are pending, sir. Yeah. So this is what we do at IOB in molecular, in microbiology per se. Yeah.
Great work. Please keep continuing that. Now we have two queries from our participants. I'm sure you will be able to help them.
Sarish
Dr. Prasanna Kumar sir has the question. Is the principle used in diagnosing other conditions apart from resistant MTB?
Yes, yes, yes. The sloppy molecular beacons we also use for genotyping when you don't have a sequencing platform and you want to know. Like usually, what people do is they do amplicon sequencing and they identify, okay, this person is having a heterozygous or homozygous or he's a wild type, which is necessary when there is single gene mutation disorders. Okay, so in those places you can use this platform sloppy molecular beacon and identify with different melting temperatures, okay, whether he is having a wild type, he is having an alternate, or he is having a heterozygous condition. Yes, there we can use. Apart from that, you can design this probe for other bacterial resistance also. We work on MTB, and the most till now the assay which is in IVD mode and using molecular beacon for microbiology is GeneXpert. That's the reason why I explained GeneXpert. Yes. So
Thank you.
what are the limitations of
Yeah.
Yeah,
Yeah,
I was
please.
just
Good
thinking
evening.
that Dr. Preeti has asked. The limitations again, even the GeneXpert Ultra misses certain mutations. As I shown in that previous diagram, there was a red mark which we had put. So that is nothing but a zone which mutation is missed. But WHO mutation catalog tells no, that is a mutation of importance. It is associated with resistance. It is not associated with resistant in TRIM. It is not insignificant or non-significant mutation. It is associated with resistant, which is missed by GeneXpert. One of our paper is coming out, and one paper has already mentioned how the previous G4 missed that particular mutation. And also, this paper will come out in another 3-4 months, where we will show how that particular mutation is getting missed. And when you're missing this particular mutation very frequently, indirectly we are enriching that mutation in the community. So yeah.
Thank you. I think you have answered the
Right this
queries.
moment, thank you, sir. Right this moment, I
Thank you,
forgot
sir.
the name of that particular mutation. Yeah, sorry, sir.
Yeah, I think Kulkarni sir wants to come in.
Yeah.
Sir, please.
Can I have 3-4 questions, short ones?
Yes, sir.
sir.
Please, sir.
Okay, I will club them together because they are virtually the same. This is about possibility of training.
Training.
Okay, like training. Say in the sense that in-house establishment of real-time PCR, how to standardize that.
Hmm.
Okay, see, means right from synthesis of primers, everything. Okay, so this is one. Similarly, about multiplexing,
Hmm.
basics of multiplexing training. Suppose we want to establish a multiplex PCR, that training. And third, again, about training on melt curve analysis. So, what are the possibilities?
On primer designing is easy. We can use any of the softwares which are available, which we can have the training. But currently, I am a student at Institute of Bioinformatics, so I don't do the trainings. Uh, this was like a part of educational program, so I was doing this. And apart from that, primer designing training, lot of training materials are available by Kayagen also. Even they give a lot of training materials, and it's basically uh, most of the time to know what you want to test, what is your genome, and also what particular platform we should use to design that particular primer. And second question, how to choose the things for multiplexing in the PCR, or how to establish a PCR setup. First thing is to have a good support. I usually tell this because I have experienced it, to have a good support from your HOD. Once that is there and you have money for getting a PCR machine, then most of the diagnostic assays which we do have already a ready-made available kits. So if you want to detect for something, for example, your particular area has some particular disease more common as compared to other areas where it is not very common, for which you don't have a in vitro diagnostic assay. For that, when you want to design, just first start with designing a conventional PCR. Design it, you have a primers, and run it, you see the bands. So once you are confident with that, then you can go ahead by designing your probes, because one single probe costs around 95,000. So we cannot waste money on probes suddenly. Once we know we are choosing the proper area, so the background work done is more than the actual lab work which we do. Even in our lab, hardly I go into lab work for 3 to 4 days a month, but most of the time it goes in designing, reading, and all those things. And for training on melt curve analysis, I'll be happy to do, sir, not a problem at all, for doing a training on melt curve analysis. But it's a very small topic. Once we have different PCR readings, we can very well do that. Uh, I'm
For,
really
for
sorry for the
example,
voice break.
just I wanted to. No, it's okay. Just what I wanted to say that bell curve analysis can it be a substitute to multiplexing?
Um, it can be substitute. No, we don't say it as a substitute, because uh, if you're synthesizing, be it Tachman probe or molecular beacon probe, you will be spending the same, almost the similar amount of money, which is costlier. So instead of designing three different probes for Tachman, for one for wild type and for mutant and the heterozygous, you design one single probe and test it by using sloppy molecular beacon. So it's not an alternative for multiplexing, but you want to do a higher number of multiplexing, like how I mentioned, we are currently doing a multiplexing of 42 plexes, 42 genes, but that is for sequencing. Again, in that case, you have to keep in mind one primer should not go and attach with another primer. It should not form primer dimers. Multiplexing, in case of sloppy molecular beacon, we do it especially in case of the genes where you get lot of mutations. So multiplexing is not an alternative for the type of probes, yeah, and the
Okay.
Medcurve analysis. Yeah.
Okay. Now, the international scenario and Indian scenario, if
Hmm.
we compare at what place we are, where are we compared to the international scenario in molecular biology as far as development of usable techniques in the field? Because now biofire is there. Okay, biofire is too expensive for us.
Mm.
Now, suppose if you are developing something like 42 multiplexing, so it is as good as a biofire.
Yeah. Okay. I'll take that
And
question in two
Indian
parts.
versions will be much cheap.
Yes, sir. Yes, sir. Definitely. So I'll take that question in two parts. There is a lot of disconnect between the clinicians and researchers in India, which usually doesn't happen in other countries. So what is happening is researchers are developing, it is not reaching the clinicians. Clinicians have a question, the question doesn't reach the researcher most of the time. And India is still in the race. We are not out of the race. We are still in the race. And also the money given in India for research is painfully very, very, very low. So whatever we have in that, we are developing good assays, but it's not of international standard because the I will tell one very practical scenario which my guide was telling. She used to design probes every day. She used to design today evening, go back home. Next day morning, she used to get that probe synthesized in Stanford because IDT synthesizing facility is there in Stanford. So she doesn't have to wait for 1 month to get her probe so that she will do the PCR. So that is one thing. That is where we are lacking. We don't have in-house synthesizing facilities in any of the institutes. We take it any AIMS, they don't have. Take it IASC, they don't have. These are one of the biggest research institutes. They don't have their in-house synthesizing facilities. So research, doing a research, I feel, but that is the fact which when I hear from her, I used to design 10 probes and it used to come next day. So I feel, oh, I have to wait for 1 month for my probe to come. That is one thing. Sorry, sir, I forgot your second part of the question. Are we there yet? Yes, we are there yet. The only thing which is needed is capacity building in India. uh, Sequencing, whenever I talk about sequencing, no, my friends will be like, oh, it might be some rocket science. No, it's just PCR. Sequencing is PCR. You have to do different kinds of PCR and then purification. So capacity building is very much needed in India, where everybody need to know how to do the technique. Then it's easy task. And coming to biofire, I completely agree, it's too costly. I myself would not get it done if it is not that much needed. For 42 plexing and all, why I was telling is that 42 plexing we do, and then we do nanopore sequencing. So in the PCR plexing, we have not reached that. General manager for South Asia of Altona Diagnostics. We were talking about how we can have different multiplexing PCRs already, making it into a cryo preserved things where they can just add DNA and the PCR is done very fastly, where they need not add enough, like add this much prime for the technicians, because a lot of places the technicians whom we have are BSc graduates or maximum MSc graduates. So we don't get a PhD to do that kind of PCR kind of work where you need to have that stability in the hand and that kind of troubleshooting ability. So uh, we
So
are lacking
basically,
in
you
all
are
those places.
discussing dry PCRs.
Yes, not dry PCRs, even for uh, any kind of assay, when we talk about multiple plexing, everybody comes with multiple tubes. When they talk about multiple plexing, they are adding different reagents into one tube. What I was mostly presenting there, or keeping my point, was it should be one single tube, but it should have multiple markers in itself. The person who is doing should not have a problem doing. That's still in uh, developmental stage. Currently, I cannot talk about it also. We have not got results for that
Thank
yet.
you very much.
Yeah.
Thank you very much for your elaborate answers. And hope you will permit us to contact you whenever we are in difficulty. Hello.
Good evening, sir.
Uh,
Good evening, sir.
I think
Good
there's
evening
some
to you.
network issue with Dr. Shruthi.
Sure.
Yeah.
Good
Yeah,
evening,
sure, sir. Sure, sir.
Sudhindra.
Good evening, sir. We are waiting for your comments,
Yeah,
sir.
actually, there was some audio problem earlier. I could not, this thing is actually, I think we should have some kind of workshops instead of just 1 hour lecture. It will be very difficult to particularly understand various this thing. I think we should have frequent biostatistic, bioinformatic workshops, actually. And hello, could you hear
Yes, sir.
me?
Yes,
Yes,
sir.
sir.
Yes, sir.
And of course, primer designing is not a problem. Recently, I am finding it difficult to design the probes, actually. Dr. Shruthi, she nicely explained various things, but probably in some other setting, she can take on how to design primers, how to design probes. Because once we design primers and probes for real-time PCR, and if you order in bulk, it works out very cheaper. Probably for 100, 150 reactions, it may cost some 25,000 rupees. Real-time PCR probes, I mean TACMAN probes and all. Hello?
Yes, sir.
Yes.
Yes, you are audible, sir.
Yeah, so for to understand many of our microbiologists, the nuances of this bioinformatics, it's better to have workshops and practice while they are teaching to design various things. It's a very nice talk. I think it stimulates a lot of youngsters. Though there are many commercial kits and systems have come like BioFire and etc. But still, they are not within the reach of the common labs and the common man. So I think indigenously, like Shruti, she told, she developed 42 plex designs. So those kind of things will come handy and very economical.
Sir.
Thank you.
And thanks a lot for
Thank you very much.
actually, and I always keep telling Dr. Shruti, as Satnana told, one of the hidden gems. There are many people like this because and our postgraduates are not getting exposure to such things, bioinformatics and this work. More and more workshops need to be organized so that
Actually, many biotechnology colleges across the country, many biotechnology, BTech biotechnology, MSc biotechnology, particularly in good universities, they are constantly working on this. I think we have to tie up with them. We had recently one workshop followed by a small CME on bioinformatics. It's very interesting. And I think we have to rope in the biotechnology experts from these colleges and conduct workshops regularly so that our young microbiologists will be immensely benefited. Once they get the grip of this thing, I think they will be much more smarter. They outsmart probably even biotechnologists, I feel.
this is a future bread and butter, going to be our bread and butter in future. So,
Yeah, that's true.
so
Molecular
over to
biology is
Satnam, sir.
going to rule because things like BioFire and all, they are made things very easy. Only thing is expensive, but anybody can perform it. Even a person need not have a degree to perform this. And instant results, highly standardized. The problem with most of these the molecular gadgets is standardization. In-house things may not work. It has to be standardized and commercialized, and then only, and with all approvals, people will accept those things.
True, sir. True.
Thank you.
Thank you. Thank you, sir.
An excellent presentation by Dr. Shruthi.
Thank you
It's
so
very
much.
nice. She simplified very nicely.
And every time we listen to Dr. Shruthi, we get more motivated to work in this field. So
Very
Thank you,
nice. Thank you.
Yeah, if there are no more queries, yeah, I would again like to thank Dr. Shruti on behalf of the Indian Association of Medical Microbiologists Karnataka chapter, the whole team of the office bearers, and we were fortunate to have the first masterclass by Dr. Raghavendra Kulkarni sir, and yeah, he's been a constant support for all the sessions. He's been attending all the sessions and taking keen interest in these proceedings. And thanks, Appal Raju sir, for also gracing the events. And yeah, we look forward to more support guidance from all the seniors. And we also request Dr. Shruti and all the hidden gems, as Surendra keeps telling, to keep be in the loop and try to motivate the future microbiologists, and also to help us get trained in all these techniques. So thank you so much, and yeah, it's been a wonderful session. Thanks a lot.
thank you, sir. Thank you so much.
So
thank you.
myself, Dr. Satyanarayana, on behalf of IAMM-KC, thank all the participants for attending this session, and yeah, we look forward to your support in the future events as well. Thanks a lot. Happy weekend.
Thank you, everyone. Thank you, sir. Happy week.